Let's see. So let's just do what we always do and go to the warm-up and kind of review the stuff that we covered last class. So again, yeah, you can see looks a lot like him, but it's not quite. His face isn't quite as round or something. So here we go. All right. Which of the following breaks down cyclic AMP or cyclic GMP to end the second messenger pathway? So this one, the phosphodiesterase, this one is going to come up in the future as well. When we look at how rods and cones convert light into electrical signals, it's all going to be about phosphodiesterase again. So you definitely want to know what phosphodiesterase is and what its role is. Again, it's the enzyme that ends that second messenger pathway. So once it starts, it doesn't just keep going on forever. And adenylyl cyclase is the stuff that makes the cyclic AMP once you start the second messenger pathway. Protein kinase A is the stuff that is turned on by the cyclic AMP to phosphorylate things and cause things to happen in the cell. The G protein is the first step once the receptor binds and is activated to kind of start the whole chain reaction. To G protein then was the thing that activated the adenylyl cyclase, which made the cyclic AMP, which activated the protein kinase A, and then finally the phosphodiesterase came in and chewed up the cyclic AMP to stop the whole thing. So you definitely want to make sure that whole kind of chain of events is clear, because second messenger pathways are kind of at the core of all sorts of signaling in the body. Give at least 2 different benefits to using second messenger mediated pathways. So this one's, a lot of people got this, but some people did not. And I'm not sure if, like, some people, when they answered this question, just, gave me, parts of the process, like, create cyclic AMP or something. And it's like, that's part of a second messenger pathway, but it's not like a benefit, So when there's a question like this, make sure you kind of go back, and I've mentioned this before, I'll mention it again. Always go back and read the question and double check that you actually answered the question that was being asked. Because I think for some people, if they would have gone back and like reread the question, they'd like, well, wait a second, this isn't a benefit, this is just a description. So what are benefits of second messenger pathways? Because they're definitely not faster or less complicated. they're big complicated pathway.
Increasing control.
So there's a lot of opportunities for increased control. You have lots of, again, we talked about phosphodiesterase. You can have pathways that turn on or off phosphodiesterase and makes another pathway more or less likely to continue on. Since there's so many kind of moving parts in a second messenger pathway, there's lots of places you can come in and tweak it. What are some other benefits?
Flexibility.
There's a lot of flexibility, right? You can have the same receptor bind the same ligand, but have lots of different ultimate results depending on how you hook up the pathway. Or like we mentioned, you can have many different kinds of receptors, ultimately all resulting in the exact same action in the cell. What was kind of funny, some people wrote like, you can have more, what is it? The same ligand can result in multiple, or different actions, also there can be flexibility. It's like, no, that is flexibility. So let's see. If we continue on, which of the following are important second messengers? So calcium, cyclic AMP, those are what people go with them. Protein kinase A is just part of the pathway. It's not officially a second messenger. Same as adenylyl cyclase makes the second messenger, but it's not. PDE is the thing that actually breaks down the second messenger. Serotonin would be what we consider a first messenger. That's the thing that's actually docking at the receptor outside the cell that ultimately results in the buildup of the second messenger in the cell. So,
Professor.
uh-huh.
So when I studied this over the weekend in the textbook, it said that there's this section that talks about secondary messenger molecules. And it says, it has 4 things listed. It says protein kinases, phosphorylated proteins, calcium binding proteins, and calcium cations. So that.
So those are part of the calcium-mediated second messenger pathway. So calcium can act as a second messenger, but some of those other things are more involved in the pathways. I wouldn't consider them official second messengers. I'll look at what she has to say. But in general, when I think of a second messenger, I think of specifically like the cyclic AMP or the calcium ions, but not those other things. I think of more as the machinery that then those use to continue on the pathway.
Okay, it's on page 181, chapter 6.
Okay, I'll take a look at that during the break. All right. Let's see. A cell whose membrane is permeable only to potassium. All right, so this one here, we're going to be spending the rest of today talking about membrane potentials. Today is all about graded potentials and action potentials and how synapses can create potentials and things like that. So you really want to be clear about what happens if you open up potassium channels in a cell. And again, in general, is potassium higher in concentration inside or outside the cell?
Inside.
It's much higher inside. So if we open up potassium channels, if you just think about the diffusion gradient, which way is the potassium going to go?
Out.
It's going to leave, right? So you're going to have positive leaving the cell. And so now we're creating this charge imbalance where it's going to be more positive outside, more negative inside, because the positive is leaving the inside of the cell, going out of the cell. So that's going to leave the cell being more overall negative. Remember, the membrane voltage we always talk about as being the inside with respect to the outside. Similarly, or I should say conversely, with sodium. Sodium is more concentrated outside than inside. So if we just looked at sodium and opened up sodium channels, sodium would come in. So now we'd have more positive inside compared to the outside, and we'd have a positive membrane voltage building up. So you definitely want to kind of keep that straight, since again, that's going to be at the core of the action potentials that we're about to see in a few moments. We're going to basically be changing the membrane potential using gated channels, voltage gated channels in this case, to make the cell primarily permeable to sodium, in which case the voltage is going to go way up, and then make it permeable to potassium, in which case the voltage is going to get negative. So you really want to kind of remember those equilibrium potentials we were talking about last time. What is true about protein kinase A? Turned on by cyclic AMP, yep. Can phosphorylate things, yep. Doesn't really do that. All right, so let's stop sharing here. So before we kind of dig into membrane potentials at a deeper level, are there any kind of questions or clarifications about the stuff we talked about last class? You know, that push and pull between the diffusion gradient and then the electrical force, which is building up in the opposite direction, so they balance out into the equilibrium. Okay. Then we will continue here. And it probably is going to make sense to kind of introduce a neuron, since a lot of these kind of different kinds of voltage changes across the membrane we're going to talk about occur in neurons and in muscle cells, but it's going to make our story kind of tie into the actual body, if you actually can put it in the context of actual neurons. So let's introduce kind of your classic multipolar neuron, which I'm sure you've all seen before. You've seen in Bio 110. If you've been in anatomy, you've most definitely seen multipolar neurons. So neuron, also called a nerve cell, also called a nerve fiber. I should just put those there. So neuron sometimes is AKA, also known as a nerve cell, also known as a nerve fiber. Fiber because they tend to have these long axons, kind of big, long, elongated cells. Multipolar because they have lots of things, lots of processes connecting up with the main cell body. Again, if you've been in anatomy, you've learned about unipolar neurons with one main connection and bipolar neurons with two main connections. But the most common kind of neuron, the main neuron that kind of makes up your brain and all that, are these multipolar neurons. So that's what we're going to focus on. All right, so cartoon-like. Let's put this thing into context. So there is my multipolar neuron. Maybe I should color it in a little bit so you can know the inside and the outside. Okay, so there's my neuron. It can be really big. Like I said, it's nerve fiber because it's kind of long. This big process that comes off of it, where messages are usually leaving, the axon. You have axons that leave your spinal cord and go all the way down to the tip of your toe. These can be 3 feet long. They might be shorter, but that's why I said fiber. They can be really long. The axon tends to be the main process that leaves where messages are coming out. The little processes where messages tend to be coming in, we call dendrites. Dendrites just means like little branches. So we have message signals coming in, the dendrites. One main kind of output. It's worth, this is actually going to be important. This little place where the axon originates is called the axon hillock. We'll come back to the axon hillock in a little bit. That's kind of the integrator, basically where all of the different input to the neuron kind of gets added up and where you decide whether or not you're going to send a message down the axon. So the axon hillock is going to be important. The cell body we just generically call the soma. Soma is just a synonym for the neuron cell body. At the end of the axon, it branches into lots of little branches. We call these the telodendria. Telo means far, right? Like telescope looks at things far up in the heavens. Television looks at things that are not happening in your house. Telodendria are the little branches that are far end of the cell here. And then each of these telodendria end in these little knobs. These are called, they can be called synaptic knobs, or they can be called boutons, means little buttons. They have the different names. I usually call them boutons because it's just fun. It's kind of, you get to speak French. So the synaptic boutons are the things that are then going to talk to other neurons. So if I have like a green neuron here with its dendrite, this little connection is going to be the synapse, where here's my green neuron, which is continuing on someplace else. The synapse, the synaptic knob or bouton will make this connection, which we're going to talk about the synapse in a lot more detail later, but it's going to make a connection with the dendrites. Or so you can have, this can be an axon from some other neuron coming in and talking to the dendrite. You can also have this would be an axosomatic synapse. This would be an axodendritic synapse. Just to kind of give you an idea, you got this thing making connections with lots of other neurons. So these little telodendria and these little boutons are then coming in and talking to other neurons, making synapses either on their dendrites or on their cell bodies. And I've drawn this, very simplified, so it's not too crazy, but a neuron in your brain might have hundreds of, 100,000 connections with other neurons doing this way. So it can get really complicated. It's connecting to tons of other things, and tons of other things are coming and connecting it to it. So I've drawn it rather simplistically here, but imagine this being kind of super complex, up to like 100,000 interconnections here. So this is kind of the basic picture of a multipolar neuron. What other things before we just leave this? One other thing I will talk about, obviously, the soma, the cell body, that's where we have the nucleus. That's where we've got all the ribosomes for making proteins and stuff. But you actually do need stuff down here on the far end. And like I said, this could be a meter, this could be 3 feet away, a meter away. So we need ways to kind of transport things. along this really long, big cell. So it's worth mentioning the idea of axonal transport. So there's slow axonal transport. This happens to be more just kind of the flow of cytoplasm. And it's, in fact, I should, what is the actual speed? It's actually very slow. Do I actually have a, oh, dagnabbit. I don't actually have the, oh, yeah, no, I don't have the actual number. Oh, no, I do have it. No, I don't. So it's slow. It's like millimeters, millimeters a day if you're lucky. And if this thing is 3 feet long, slow axonal transport is not going to get you from one end, from the cell body out to the telodendria. So fast axonal transport is more like a railway system. It's actually got microtubules. It's fast. It has microtubules that run along the length of the axon. There are special little motor proteins, these little kinesins that basically walk along the microtubules. Kinesis, not kinesin. Basically, you power them with ATP. Again, if you can actually find videos of this where they'll attach a little glass bead onto the kinesin and put it in a thing with microtubules and puff ATP at it, and all of a sudden you'll just see it walking along. So this can go pretty fast and far, like tens or hundreds of millimeters a day. So if you need to get something from one end of the cell to the other end of the cell and don't want to wait forever, there actually is a way to move things. And two more words you should know. Anterograde means moving forward. Retrograde means moving back towards the cell body. We're going to talk about this in the propagation of action potentials as well, but this transport can be anterograde, moving towards the end of the cell, or retrograde, coming back. Part of the reason I want to bring this up is it actually becomes relevant in pathology of, for instance, herpes. So if you get herpes, you get infected with herpes, you get sick. Most people know if you get herpes, you got your initial presentation, you got some flu-like symptoms or whatever. You got, like, depending if you have oral or genital, you've got these lesions showing up around your mouth or your genitals, and then it goes away, but then it's never really gone, right? You can have it flare back up due to stress or whatever. All of a sudden, you've got a flare up again of this thing. So it obviously, the virus didn't disappear. It just kind of went into hiding. And the place it goes into hiding is in your neuron cell bodies along your spine. So basically, you have your dorsal root ganglia are the cell bodies along your spine for your sensory neurons, and they reach out to where you're actually having your receptors for, along your face or along your genitals for feeling stuff. And this little virus can hook a ride in the retrograde axonal transport and set up camp. And so even after the infection is done with its initial presentation, the virus is still kind of camped out. And for whatever, I'm not sure even they know exactly how it happens, but it can hook a ride back on and make it back out to the periphery. And all of a sudden you've got a flare up again because now the virus is up on the surface again. because it's got this nice little railway to go back into its little safe hideout and get back out to your skin. So it's kind of fascinating. Okay. So that is the kind of anatomy of the neuron to the degree you need to know it for now. And now what we're going to do is we're going to talk about different kinds of voltage changes that happen across the membrane of that, and where they happen and how they happen. And let's do that. So I'm going to start with the idea of graded potentials. Versus action potentials. And again, everything we're talking about today will be on your exam on Tuesday. Remember, your first major exam is 1 week from today. It's going to include everything from the first day of class, like homeostasis and chemistry review, up through the stuff we're doing today, which is basically talking about graded potentials, action potentials, and then also the synapses that are related to kind of create these or create them. So, and then once we're done with this, we'll have all the background we need to finally start our first system. We'll actually start the nervous system next time. We'll finally have kind of got the chemistry background, the basic cell physiology background, and we can start putting things together and look at the bigger picture. So graded potentials and action potentials. Again, potential, this is a synonym for membrane voltage. But that's what we mean. The potential, electric potential, or membrane voltage is the same. We just use them interchangeably. So let's start with graded potentials. Graded potentials basically happen when you have ions cross the membrane. So they happen when ions enter or leave the cell. Depending what kind of ions they are, the voltage might go up or down. Right? If you have potassium leaving, maybe the voltage is going down. Or if you have chloride coming in, the voltage would be going down. Or if you have sodium coming in, the voltage is going up. Right? So it can be positive or negative depending on what kind of ions are entering or leaving. Can be small or large. What we say is continuously variable. So they can, if you have a lot of ions moving in, maybe you'll have it go up quite a bit. Or if only a few move in, it will go up a little bit. And you have a whole range. So continuously variable means it can be anything from a little to a lot in a continuous range. And it can be positive or negative, depending on whether things are entering or leaving and whether they're anions or cations. So continuously variable, positive or negative. They are local, meaning wherever you're opening channels and letting things move in or out, that's where this graded potential is going to be. So localized on the membrane. Wherever you are opening the channels, that's where the voltage change is happening. and they tend to be short-lived. Basically, once you have restored the membrane permeability, then this graded potential kind of fades away. Right, so you're basically, usually you're opening channels, opening or closing channels. Ions enter or leave the cell. The voltage on that little area, again, localized, can go up or down, small or large, tends to be short-lived. So that is graded potentials. Action potentials. Oh, and I should mention just where they are. Graded potentials, these tend to be like on dendrites and on the soma of the neuron. So action potentials happen along the axon. These are going to use special voltage gated channels. We're going to look at this in much more detail in a few moments. There's going to be a voltage gated sodium channel and a voltage gated potassium channel that work together to create the action potential. These are all or nothing. There's no positive or negative. There's no small or large. They're either going or they're not. A good simile is, it's kind of like lighting a fuse. Right? If you've got like a firecracker and you light the fuse, it's just going. It go, you can't like light a fuse negatively or positively or strongly or not or weakly. It's you either light it and it's going or it's not going. That's what I mean by all or nothing. Or another like lighting a fuse or like pushing a domino. Right? If I have a bunch of dominoes and I push over the first domino, each domino is going to whack and knock down the next domino. It's just going or it's not going. You can't make it go stronger or weaker or negative or positive. It's either the domino chain or whack or knocking each other down or it's not. Right? So that's what I mean by all or nothing. It's like it's kind of waiting to go. And once you trigger it, it then is going to run, and it runs along the whole length of the axon. So rather than being localized, travels long distances. Right? And is, I should say, and long-lived. Right, so if I go back to this picture here, my graded potentials are going to be happening, let me maybe do some color. Graded potentials are coming in here, but then once they happen, they go away. There's some graded potentials over here, but then they go away. Action potential, once an action potential gets started, it just keeps going and doesn't stop till it gets all the way to the end. And again, like I said, this could be 3 feet long. So it's this chain reaction thing that gets started here at the hillock, but then doesn't stop till it gets to the end. So the action potential travels long distances and lasts as long as it needs to get to the far end of the axon here. Whereas the graded potentials, you open up some channels on the membrane, the voltage goes up or down a bit, and then it resolves and goes back to where it was. So any questions about the core difference between graded potentials and action potentials, theoretically? Again, for graded potentials, what we're going to find is all we need to do is open and close channels on the dendrites in the soma. Right? If you open up a sodium channel, the voltage will go up a bit as sodium comes in. If you open up a chloride channel, the voltage will go down as the anions come in. So we're going to see graded potentials happening, like, when I was talking about these synapses. Usually when a synapse is fired off, what happens is you ultimately open channels that create these kind of local graded potentials that ultimately are going to affect whether or not the axon hillock hits its threshold to fire off an action potential. So we'll talk about that much more completely in a few moments. But what we're going to do now, again, graded potentials are pretty simple. Basically, you open up a channel, ions come in or out, the voltage changes. That should feel rather intuitive, given that voltage is just charge separation across the membrane. If you alter that balance of charge, you change the voltage. Action potentials now. And we usually abbreviate AP, action potential. So for action potential, you need to get to know two specialized voltage gated channels. So actually, I'm going to use my little typing keyboard to make this faster. All right. So in the axon of the neuron, there are these special voltage-gated sodium and potassium channels that are going to be responsible for creating and maintaining the action potential. And so I'm going to introduce them. Okay, one. So the slow voltage gated potassium channel, this basically opens if V sub M is greater or equal to minus 55 millivolts. Slow takes around 1 millisecond to open or close. So basically, so remember VM, what is the voltage of a resting neuron we mentioned last time?
Around minus 70.
Exactly. So a neuron at rest is around minus 70. this threshold here is right here. So as long as the cell stays at its resting voltage, these little voltage gated channels are going to stay closed. The reality is the cell is at rest, but it's kind of going up and down and up and down. If at any point the voltage is above that minus 55, they'll open. They don't open immediately. They're, again, I always think of them like in those old like cartoons. Which way did he go, George? It's like, you know, the voltage has gone over the threshold. It's over minus 55. These voltage gated sodium channels are like, oh, look, we've reached threshold. It's time to open up and let potassium cross. But it takes about a millisecond before they actually open up, which is going to be important. Once the voltage goes down, back down, they close. But again, there's a lag. It's like, oh, look, we're down below threshold again. I guess it's time to close, right? So it's going to matter that these things are kind of pokey compared to, you'll see the sodium channels are fast. They basically snap open and snap closed when they do their thing. So this is the simpler one, the potassium channels. They basically, like I said, they'll snap open, or they'll open when the voltage goes above this threshold of minus 55. They'll close when it drops back below. But there is this kind of pokiness, this lag to them, both in terms of opening and closing. It takes about a millisecond for them to actually do their thing. The other ones are kind of more complicated, the voltage gated sodium channels. So these also open when V sub n is greater than equal to minus 55 millivolts. So they have the same threshold, but they open very quickly. So these are fast. Once the voltage goes to minus 55 or above, they basically snap open and they let sodium in. But then they have this in fact, let me maybe draw a picture here. Let's say this is my cell membrane. Here's my voltage gated sodium channel. They inactivate after 1 millisecond. Even though VM is still greater than minus 55. So the basic idea here is, let's say the membrane voltage is going blah, 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 blah. Here's our threshold. The membrane voltage gets above here. These things, we've hit threshold. Sodium rushes in because these things have opened. But after 1 millisecond, there's this other part that just blocks it. And so once you trigger these, they open quickly, but inactivate after 1 millisecond. So this is going to be important. It would be, I don't know, it's a metaphor, simile. Like, yeah, some door, it's like, the door has like some little like hydraulic thing. So, whenever the door opens, it automatically, right? So whatever triggers it to open, but then after a millisecond, it closes on you until you manage to trigger it again. So does this make sense? Are there any questions about the dynamics of these fast sodium channels? If the membrane potential gets to minus 55 or above, they'll open quickly, but after a millisecond, they will stop conducting. And you have to bring the whole membrane potential back below minus 55 for everything to reset and be back to normal.
I guess I have a question, but it's going back a little bit. So when you were talking about graded potentials, you said it can be positive or negative, and then the action potentials are not positive or negative.
Right.
So, I mean, there's a lot of obviously like ion exchange going on, but does that just mean the whole, like the voltage of the cell is not changing with the action potentials?
So it's going to be clear in just a few moments.
Okay.
Yeah, so there are a variety of these different local potentials happening to create the action potential, but the action potential itself is like a wave that's going to continue along. So
But it's charged necessarily like the graded potentials are? Is that the
I mean, so it's still made up of ions crossing the membrane and voltages going up and down, but the action potential that it's kind of like if you think about a wave on the ocean, yes, you can think of individual water moving up and down, but that's not the thing you're actually paying attention to. What you're interested in is this kind of meta thing happening of a wave moving rather than of water molecules moving up and down in a local area, right? Like if I'm on the sea, there's a wave moving, and the wave's a little farther, the wave's a little farther. We have water molecule that went up and down, went up and down, went up and down, but this water molecule is now just continues sitting here. This is sitting here, but the wave is continuing on, and the wave is what I'm in. The action potential is like a wave, even though there is kind of local complications happening with ions moving in and out and voltages going up and down. Overall, the wave is going to continue moving from one end of the axon to the other. Right? If I was just sitting in my little boat here, as the wave came by, I would just go up and I'd come down, and then everything would be still. But if I have kind of this more broad view, it's like, oh my God, look, there's a wave traveling across the sea. Right? But if I was just sitting in my little zone here and didn't care about the big picture, I would just say, oh, I went up and I went down, and then it stopped. Right? So that's kind of action potential is like the wave. It's this kind of more meta thing that is getting created and continues along. And I will show you how that works right now. So let's get back to our neuron. There was the hillock. And remember, the axon is full of these voltage-gated sodium and potassium channels. So let's start right here at the hillock. And then we're going to carry on our thing for the rest of it. So let's look what's happening right there. We're going to look at V sub M. So this is that threshold. We need to pay attention to that because if the voltage gets up to there, we know that these voltage gated channels are going to get triggered. And then, like we said, voltage is typically around -70. What's happening though is, there's all the synapses talking to this neuron. And there are graded potentials happening on this neuron. And some of them are positive. We have so things are actually like pulling the voltage up, pulling the voltage down based on all the synaptic activity and all the graded potentials across here. So the reality, the voltage, as we're looking at it here at the hillock, which is where we're paying attention, is actually jiggling up and down. And let's assume at some point, bing, it hits that threshold. What is going to happen the moment the voltage in the membrane, and again, we've got these special voltage gated channels sitting here. What's going to happen as soon as the voltage of the membrane hits -55?
They're going to open and then close a millisecond later.
Well, in which particular one are we going to notice first?
The Na channel.
Exactly. Remember, the sodium channels are the fast ones. As soon as this happens, like I said, the potassium channels are like, oh, look, something's happened. We should start opening. But the sodium channels instantly snap open. So sodium channels are going to snap open. What is going to happen as soon as all of a sudden the cell membrane is primarily permeable to sodium? We have lots of sodium channels open.
Then they're going to close.
Well, what's going to happen to the voltage of the membrane though? What happens if you have a cell that is got a major sodium conductance? What is the voltage going to look like?
Positive.
It's going to be positive. Remember, it's going to go up towards that equilibrium potential for sodium. You've got all this sodium rushing in. So we got this big rising edge of this, what's going to be our action potential. The voltage goes up plus 40 or 50 or something as all the sodium rushes in. Right. This is why I was saying it was important to know the equilibrium potential for sodium was around plus, I said plus 60, but it was close to this. Let's put on our milliseconds here. After about a millisecond, what's going to happen? Two things are going to happen.
It's going to close.
Yeah, so basically the sodium channels inactivate. So the sodium channels are going to inactivate after a millisecond. And then what else is happening after a millisecond?
The potassium channels open.
Exactly. Now the potassium channels are open. So all of a sudden, the sodium channels have been activated and the potassium channels are open. Which means now we have primarily a potassium conductance, which is going to bring us down towards minus 90. Right, so that's going to cause this major falling, not just because we have more potassium channels than normal resting, we actually are going to be down below minus 70. There's this undershoot. Right, because it's even more potassium conductance than normal. So it's going to be even closer to that potassium equilibrium potential. And then after another millisecond, the potassium channels resolve, and now we're kind of back to normal. So this thing that I just drew here is kind of going up, coming down, undershooting, and then kind of being back. That is the action. That is an action potential. Let me mention a few more things about this, and then we're going to put it in the bigger context up here, because obviously this is just the beginning. Absolute refractory period. So 1 millisecond after this thing fires off like this, it takes about a millisecond for the channels to reset before this can happen again. That is going to be important. We'll see in a few moments why it's actually important that once the channels go open and close, it takes about a millisecond before you can actually have another action potential on that same piece of membrane right there. So absolute refractory period means you have to wait at least 1 millisecond after you fire off an action potential before you can have another action potential on that same piece of membrane with those same channels. There is also something called a relative refractory period, which I'll mention just for completeness. That is while you're still in this undershoot, while the voltage is still lower than normal. Remember I said this undershoot brings the voltage of the membrane lower than the normal resting, which means, you'd have to use more stimulation to actually bring the cell back up to the threshold. If you wanted to fire, if you're in the relative, let me say this better. If you're in the absolute refractory period, nothing you can do can make the membrane hit another action potential. You just got to wait till the absolute refractory period is over, about a millisecond. If you're in the relative refractory period, the cell can fire off an action potential, but because it's more negative than normal, it takes more stimulation than normal to get it up above the threshold. So relative refractory period, it's possible to fire off an action potential, but it takes more stimulation than it would otherwise. So, are there any questions about this basic picture here? All right, so what we're going to do now is remember everything we've been describing here. was happening right here at the axon hillock. As the membrane at the axon hillock went over threshold, all of a sudden we started this whole chain reaction, or this whole thing, and it got all positive here. But now, again, these voltage gated channels are along the entire length of the axon. So as this little part of the membrane becomes positive, that means this next little section is now coming up above threshold. Which means what? What happens as this next little membrane piece of the membrane starts going up above threshold, as this action potential is happening right at the hillock? So the membrane potential here is starting to get positive because of all this activity. What's going to happen to the voltage gated channels in the membrane there? What happens to a voltage gated channel when the voltage goes up above -55?
It opens.
They open. So all of a sudden, the sodium channels on the next little part of the membrane open and the sodium rushes in. And the voltage goes up there and we have an action potential happen there. But then when that's happening, that means the little membrane on the next little section is now becoming more positive and goes over threshold. And what's going to happen at that point?
It activates.
It's going to open the channels on the next little part of the membrane. And they'll, maybe people obviously aren't getting an intuition about this. Maybe I'll draw a bigger picture or something. This is like really critical, that this is just feels like, oh yeah, that totally makes sense. Again, here was that. Here's my axon. Here was that hillock. Again, initially the voltage here got high enough, sodium rushed in, and we had the action potential. But as that's happening, the next little part of the membrane is becoming more positive, which means the sodium channels there are going to open, which means sodium is going to rush in there, which means now this part's going to have that action potential happening. But as that's happening, the next little part of the membrane is becoming more positive, and it's going to open up the sodium channels there. The sodium is going to rush in there and create the action potential is now going to be over here. And then as this is happening, now the next little part of the membrane is getting more positive, which means the sodium channels there are going to open. The sodium is going to rush in, and now we have the action potential forming there. So this is just going to keep moving along, like I said, like a wave. Again, wherever the action potential is at one moment, the next little part of the membrane is going to become more positive, and it's just going to keep moving along. Because at each point when it's making that little part of the membrane positive, that's going to trigger the next little part to have its voltage gated channels activate. So this action potential just propagates. Again, like lighting a fuse. Once it's started, as one little part of the membrane gets more positive, that's going to open up the sodium channels and make the next part more positive, which makes the next little part more positive, which opens that sodium channels, which makes that more positive. And it just keeps going and going and going until you get to the end. So does that make more sense? So while we're here, we might as well, let's bring in the absolute refractory period. So in my picture here, like I said, here the action potential right now is here. And it's making the next little part of the membrane positive. And then sodium is rushing in here and making the next part of the action potential as it's moving along. How do I make sure that I don't just reactivate the part of the membrane where it was just coming from, right? Because if this is positive here, yes, it's going to excite the part of the membrane further down the axon, but it's also going to be making the part of the membrane, before it also positive. So what keeps the action potential from starting to just trigger itself going backwards again?
Is it the fact that it's in the relative refractory period, so it would need a higher charge this time to reactivate?
Yeah, or not even relative, it's in the absolute refractory period.
Okay.
Right? So what happens is, maybe let me, action potential. Positive triggers the next little part to have what? When this is happening, it's now triggering the next little part of the membrane to start its action potential. But this part over here that was happening, that made this happen, this is still in its absolute refractory period. So it doesn't matter how much positive is here. This part is in rest. This part can't have another action potential yet. And by the time this is over and this is ready to go again, this excitation is far enough away that it's not going to cause a problem. So this refractory period, this ensures, anterograde transmission. Anterograde propagation. Right, because of this absolute refractory period, whenever part of the membrane is experiencing an action potential, it can excite the next little part of the membrane to have its next continuation of that excitation. But this way is blocked. It can't go backwards because this part of the membrane is still in its refractory period and is not excitable. So it can excite further this way, but it's blocked from heading back and returning back to whence it came. So that makes sense? Okay. So any questions about this core story about what an action potential is and how it propagates? So this kind of chain reaction. It starts at the hillock here, where we have the first action potential due to going above that threshold. But once you get the first one happening, it just keeps exciting the next little part of the membrane and the next little part and the next little part. And it's like, again, it's like lighting a fuse, and it doesn't stop till it gets to the end.
I actually have a quick question. So when you were mentioning like retrograde and herpes, how would is that just like a completely different thing than an action potential in terms of like,
It's completely different. That is
Okay.
a mechanical kind of railway system along microtubules.
okay, got it. Thank you.
There we're using the words for forward and backward, but it's a different, a totally different thing.
Okay, thank you.
Yeah, and the way people, I should mention Hodgkin and Huxley. Hodgkin. Oh, God dare, gosh darn it. You know, you might wonder, how did we figure this out? This was like back in the 50s. These dudes used what is called the Squid giant axon. So it sounds like it's from a giant squid. It's not. It's actually, if you just get your classic calamari squid. And most people have had calamari and beer. It's definitely worth doing. So you have your squid, and if you try to catch a squid, it's gonna go and jet away from you really fast. So it wants to have really fast transmission from, if it notices it's in danger, to being activate its muscles to jet away from you. And there are different ways to increase the speed of transmission of the action potential along an axon. So you can increase the action potential speed by increasing the diameter of the axon. This works because it's kind of like having a wire that has high resistance and low resistance. The signal stays high much longer. You don't have to keep regenerating it if you have a much larger diameter. Even in our bodies, we have some axons with larger diameters, smaller diameters, depending on how fast we care. We're going to see in our cells, we also have myelin, which is going to play an important role for increasing speed. In squids, they make a really fast signal just by having that's why they call it the squid giant axon. In the squid, it's like literally a millimeter in diameter. It's huge. So what they were actually able to do was take this giant axon from the squid, and then they would check it out. They would put different types of ionic solutions inside and outside, and then they would take a little squeegee and they would squeeze out the inside of the thing, and then they use a syringe and fill it with some other ionic solution and test that. And so they would just keep checking out, how does this thing respond? What are the electrical properties? It has this kind of ion or that kind of ion inside and out. And that's how they actually came up with all of this idea about these voltage gated sodium channels, and where's the other one? The voltage gated potassium channels. So they figured out that it had to be these things, and they actually figured out the dynamics, like the opening and closing and the speeds. And they actually had a model. Like now we can just do this on a computer and you can just watch it happen really fast. They literally, it's kind of wild. This was before computers. They literally did numerical integration of their model of how these sodium and potassium channels open and close. They spent an entire winter using adding machines, like kind of validating their model using numerical integration to show that their understanding of these channels actually resulted in an actual action potential like they observed in the actual axons. So it's kind of trippy. So they were able to actually figure this out already, like 70 years ago, using much more. Now we have so many amazing tools to look at this stuff and to model it and do calculations. But even back then, they were able to figure out a lot of cool stuff. Um, so we have a system now where you have the action potential here, and it excites the next little part of the membrane. So now with the action potential here, which excites the next little part of the membrane. So now the action potential is here, which is like the next little part of the membrane. So now the action potential here, which is like the next little part of the membrane. Then it's here, and then the next little part of the membrane. And then it's here, and it's the next little part. And then here, and it's the next little part. And then it's here, and it's the next little. And then it's here, and it's the next little part. And then it's here, and it's the next little part. And it's here, and it's the next little part. And it's here. And so this can take a while if you're just going this way. This would not be a good way to get a message from your brain to your muscles, or from your eyes to your brain, and from your brain to your muscles to say that I'm in danger and I need to respond quickly. Like your slower axons, slower transmission can be around like 3 to 5 meters per second, which is really pretty slow. Like imagine, it's like I touch a burning stove and it's like to my brain, the brain saying, oh, my hand is like disintegrating in the fire. And then I better do something. I send a message down to my muscle to pull away. By the time you've done that feedback, that kind of reflex, your hand has been incinerated. So we need to have ways to transmit messages much faster along axons. For a lot of stuff, this is good enough. There are a lot of signals that you don't have to have super fast, in which case, sending messages 3 to 5 meters per second is totally cool. But for particularly a lot of kind of sensory and skeletal muscle stuff, you want to send messages much faster. You can get speeds, over 100 meters per second using myelinated axons. So let me describe a myelinated axon and why that allows you to speed things up, and then we will take a break. We'll talk a little bit more, probably spend another 15 minutes just kind of tying it all together with synapses. So we're just done. And then we'll get into the lab. So, but first, let me spend a few moments talking about myelinated axons. So the reason why it's slow in this case is you're creating an action potential here, but it doesn't like spread out very far. And so you need to kind of regenerate it in the next little part of the membrane so it doesn't die out. And the reason why the action potential doesn't spread out very far is basically because, this is sitting in salt water. Imagine I have a battery. Now here's just your battery, and it's connected, I don't know, to some light bulb. So here's some circuit. The electrical charge goes from one part of the battery through the light bulb and comes back here. So this should feel kind of intuitive. This is, a flashlight. Now imagine these wires, and imagine we put this in salt water. And now the electrons are leaving the battery, and it's like, they're just going to short circuit. They're not going to go all the way to the light. They're just going to go through the salt water and find the shortest way back to the other side of the battery. right? So if I actually want this flashlight to work in salt water, what do I need to do?
Insulate it.
I need to insulate it. What I do is I take plastic and I insulate my wires. So the electrons don't have a choice. They have to stay in the wire. They can't go and take a short circuit through the salt water. So myelin is like an insulation for your axons, and it keeps the voltage up higher and keeps the voltage from an action potential from kind of coming back down as quickly. So let me show you how this works. So the myelin is in, they call the myelin sheaths. We'll talk about this in more detail later, but it's basically made out of wrappings of specialized cell membrane. It's kind of like a fatty jacket. There are little gaps. These gaps are important. This is called the node of Ranvier. We find those voltage-gated sodium and potassium channels here. So the way this is going to work. So this is the voltage on the membrane, and this is, my axis is actually just the physical distance along the length of this. So let's assuming, here's the hillock. Let's assume we have an action potential get started there. If it's just a regular, if there's no myelin, the action potential happens. We need to have channels on the next little part of the membrane before this drops down too far in order to make sure that the signal doesn't die, right? So if there is no myelin, you need to have channels on the next little section so we can keep it going, and channels on the next little section to keep it going, and channels on the next little section to keep it going. If there is myelin, though, in this case, it goes up, then it comes down much slower. That insulation keeps the voltage from dropping so fast. It does still drop, but it means that you have a lot longer before you have to regenerate again. So here we have the voltage gated channels in the node of Ranvier. Those are going to restore it, and it's going to drop. So wherever we have a node of Ranvier, we kind of regenerate the signal and keep it going. But notice what's going on here. And I should also mention, these nodes of Ranvier, these are about 1 millimeter apart. So pretty far apart. So instead of having to regenerate, like go, 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 go. That would be without myelin. Instead, it's like boom, 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 boom. It's just jumping from node to node, happen way faster. They call it saltatory conduction because it's like the signal just jumps from node to node. Saltaire is to jump. Right, so again, again, without myelin, it's regenerate, 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 regenerate. Every little piece of the membrane has to have channels that open to make sure that the signal doesn't die off and makes it to the very end. Again, it takes a while. With myelin, boom, 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 you're there. You only have to regenerate it every millimeter or so, instead of like every little piece of membrane. So does that make sense? What's going to happen? Like some MS, multiple sclerosis, is a autoimmune disease where you have your immune system attacks your myelin in your central nervous system. What kind of symptoms are you going to get with a demyelinating disease like multiple sclerosis?
Loss of motor function.
So basically anything that relies on transmission along these neurons. There's what are the two possibilities if you start losing myelin? What's going to happen if we don't have the myelin on an axon?
Action potentials are going to be slower.
Yeah, so one thing is they could be slower. In fact, the neurons will often start compensating by increasing the number of channels to try to keep the signal up, right? Because remember I was saying, normally the myelin keeps the signal up so you don't need channels as often. But if you're not having as much myelin, then you're going to need more channels in the membrane to keep the signal from dying. But it means that the signal's slower getting to where it used to be. So both, and again, this is both sensory and motor. Both sensory and motor signals are going to have trouble either taking messages in or controlling things. And worst case, you might have the signal just drop off and it never makes it off all the way to the end. In which case you start just getting total paralysis or blindness or deafness or whatever, because either sensory messages never arrive, or the motor signals never make it to the muscles. So worst case, it can just be, complete loss of sensory or motor function. So just kind of putting that out there, like diseases that affect myelin sheaths can be catastrophic. We'll talk more about MS a bit when we get to the immune system. And it's really different in different people. Sometimes it's just kind of annoying and it gets worse and gets better and worse. Some people, it just gets worse and worse until they die. And nobody seems to understand actually some of the dynamics of why it starts or how to control it. So it's a pretty scary thing. Okay. On that note, let's take a break. It's 9:43. Let's come back at 10:15, and I mean 10:15. It's about a little over a half hour from now. We are gonna just finish up neurons by talking about those synapses in a little more detail. So what we're gonna do now is spend a little bit of time just looking at the other main ways that neurons are sending messages and talking to each other. So a big way a neuron sends a message is with an action potential along the axon. But then the other way that neurons talk to each other is synapses. So let's look at this. So All right, so we have one neuron. Here's the black neuron. It's axon. It's going to talk to another neuron, maybe the red neuron here. So there are neurons that actually have direct electrical connections. It does happen. If I wanted one cell to be directly electrically connected to another cell, how would I do it?
Gap junction.
Exactly, gap junctions. So some neurons actually do have connections to other neurons with gap junctions, and there's a direct electrical connection. That's not the most common way that neurons talk to each other. So usually we have these chemical synapses. And the cells don't actually touch. So you'll have, if this is my presynaptic axon here, here's my little bouton, my little synaptic knob at the end. Yeah, it's supposed to be here. Let's say this is my postsynaptic dendrite here. And again, I can right, so everybody kind of has it. I'm looking right here at the place where one neuron is talking to the next neuron, and they don't actually touch. There's actually a little gap in between. So this we call the little synaptic cleft. And it's somewhere around like 40 nanometers. So what is a nanometer when I say nanometers?
It's 10 to the minus 9 meters.
Exactly, it's 10 to the minus 9th meters. One billionth of a meter. So it's a very small distance, but they're still not touching, which is important. And instead, there's going to be a chemical message that actually diffuses across that little gap. And that's going to be, basically, we're going to have neurotransmitters, which are stored in these little vesicles in here that are going to diffuse across and are going to bind to receptors sitting on the other side. So some neurotransmitter is going to diffuse across. In fact, I can use my typing tool to make this clearer. Heck. So again, it's a little non-intuitive. You have a actual chemical signal that is going to diffuse across, and at distances this short, diffusion actually happens very fast. So we're going to look at this in a little more detail right now. Like what exactly is happening? How does this work? And how does it connect with the story we've been telling up to now? So let us look at that. I'm looking at the time too. We're okay. And again, my post-synaptic, let me make it smaller. All right, so is everybody kind of oriented what we're looking at here? All right, so then we have to add in some other parts to this. And we're going to have what we call synaptic vesicles, which are basically little membrane-bound vesicles that have signaling molecules. The signaling molecules that the neurons use, we call neurotransmitters. I'm going to give them a specific shape so they can look like something that will match the receptor. So these are my synaptic vesicles. Obviously, we're going to need receptors on the postsynaptic membrane that will bind that. So we'll put those on there. These are my receptors that bind the neurotransmitter. And the one other piece I'll add in here is voltage-gated calcium channels. All right, so now we're set. And yeah, actually, just to make this even more clear, again, here is my presynaptic axon. And so you have to kind of go back to our story about the action potential, right? We had, here was some neuron and some axon. And here's the little telodendria and the little bouton. And here I'm looking at my little synapse. Some action potential is zipping down the axon. And that action potential makes it all the way to the very end. So now we go to our synapse here. So that action potential has made it all the way to the very end here. When that action potential reaches there, that's going to trigger these voltage-gated calcium channels. So maybe I'll write it out specifically using my typing tool. And this triggers the exocytosis of the neurotransmitter. So basically, I'm not going to, there are other other things involved here. There's a whole other story. We're not going to get into the more complicated story. We'll just say when the calcium comes in, that is the signal that causes our synaptic vesicles to fuse with this membrane, and the neurotransmitter ends up in the cleft. So trigger, this is really, I could say the release of the neurotransmitter. So that part makes sense. The action potential reaches the very end of the axon, and then these little boutons, these little knobs, release neurotransmitter into the cleft. Obviously, the neurotransmitter drifts across. Neurotransmitter is going to bind the receptor. On the postsynaptic membrane. And then what happens? Right? We had the action potential, which then caused the release of calcium and the exocytosis of neurotransmitter, which then drifts across and binds the receptor. What's going to happen now on this next neuron? What is the response to binding the receptor going to be? What do you think?
Would it open something? Like open a channel?
Exactly. Everybody hear Grace? When the neurotransmitter binds the receptor, this is going to open channels, open ion channels. Right, so then all of a sudden ions are going to cross this membrane. Which is what happens when ions cross the membrane?
Change the voltage?
Changes the voltage. So it's going to create voltage changes on this postsynaptic membrane. So we had an electrical signal, the action potential, which then got converted into a chemical signal, that neurotransmitter that diffused across the cleft. But now it's turning into an electrical signal on the next neuron again, because we're opening ion channels, which are causing graded potentials. And is the voltage going to go up or down? Is it going to be excitatory or inhibitory when we open channels on the next neuron?
It could be either.
Exactly. Excellent. Excellent. So can be excitatory, VM goes up, or inhibitory, VM goes down. It depends on what kind of ion channels you're opening. If you opened a sodium channel, the voltage is going to go higher, become more positive. If VM goes up, we say that it's called depolarization. Depolarization. When VM is going back down, we say it's kind of repolarizing. If actually VM goes below resting, we say hyperpolarization. Just some words for you. So back to this idea of the synaptic potential then. Here was the synapse. We released the neurotransmitter, binding onto the receptors. We have what we call EPSP, and that is going to be excitatory post-synaptic potential. Or the other thing we have are inhibitory post-synaptic potentials. This IPSP. That's if, VM goes down. Excitatory is if VM is going up. So the little voltage that happens here, the little voltage change, VM is going to go up or down based on what kind of ions are crossing the membrane and which direction. So we might actually make it more positive or more negative there. Like I said, examples, like if we let sodium in, then it would be going up. If we let chlorine in, then it would be going down, right? So it depends what kind of ion channel is being opened by this receptor. Next, on Thursday, I'll talk more about specific kinds of neurotransmitters and receptors that are typical. And but for right now, let's just say, you've got a whole, there's like over 40 or 50 known neurotransmitters, and they can do different kinds of things. Sometimes they make it more positive, sometimes more negative. So we now have everything we need to put the whole picture together. We have this idea of how one neuron is influencing another neuron through these synapses. And we also know about how action potentials get triggered once the axon hillock goes above that minus 55 millivolt threshold. So let's put it together. Here's some neuron. Again, that hillock we said was going to be important. And then you have all these other neurons coming in and talking to this one. Like I said, there were axodendritic synapses, there's axosomatic synapses. So there's all this, and like I said, an actual nerve cell might have 100,000 interconnections. But there's a lot of stuff that connecting to this thing, and then it's going off and talking to lots of other things as well. And now imagine each of these synapses is either an excitatory postsynaptic potential bringing the voltage up, or inhibitory bringing it down. And it might be going or it might not be going, so it's not, and then depending on how far it is from the hillock is going to depend on how much it influences the hillock. But what we do is we look at the VM at the hillock. And it's going to be this is summation, right? The big sigma is just the summation, adding up. We add up the total of all the EPSPs and the IPSPs that are occurring all over the cell. right? You have all this electrical activity happening from all of the different neurons that are talking to the black neuron. There's some sum total of how much excitatory and inhibitory stuff is coming in. And at the hillock, if we look at VM at the hillock, again, it's typically around minus 70 millivolts. But then we also talked about there's that threshold at minus 55 millivolts. As the voltage kind of goes up and down based upon all of this activity of all these synapses talking, if that overall activity hits minus 55, bazing, we get an action potential. And then once that action potential starts there, it's going to go all the way to the end and then influence whatever this neuron talks to. So you can kind of think of the neuron kind of as an integrator. Right? It's kind of adding up all of this electrical activity that's talking to it, both the positive and the negative. And depending on that, if there is enough net positive to take the axon hillock over minus 55 millivolts, that is going to be the trigger for this thing to fire off an action potential down its axon and influence the next stage of the neural circuitry. Right, so this is just summation of sigma. So does that make sense?
So is the stimulus is greater in the axon, the greater potential, right?
Say
So
it again.
it depends on the stimulus, right?
It depends on the sum total of all the different stimuli that are coming in and talking to the neuron.
Okay.
Right, and some of those things are actually making it more positive if it's an excitatory postsynaptic potential. Some are actually pulling it down, these IPSPs. In fact, if this is a good time, is that like, let's talk about Valium, Xanax, right, all the benzodiazepines. Right, most people have heard of the benzos. Valium is with alprazolam, and usually they have an am. This is diazepam is Valium, alprazolam is Xanax. All of these, lorazepam. There's all of these benzodiazepines that people. Why do people take benzodiazepines? What do they do to you?
They're a sedative.
Yeah, they're sedative. They're like anti-anxiety. And all of these increase chloride conductance. At the synapse. So when you take these drugs, and we go back over here, you are going to be letting more chlorine in. What is that going to do to the postsynaptic voltage if you let more chlorine in there?
Make it more negative.
Make it more negative. So, which means we are increasing the amount of these inhibitory postsynaptic potentials, which means when you get over to this picture, we're increasing the amount of negative in this sum, making it harder to bring this thing over its threshold, meaning you're less likely to have this neuron get excited and fire off something. So your whole brain just becomes less excitable. All the neurons are a lot trickier to push over threshold because you're increasing the amount of IPSPs by taking a benzodiazepine. Does that make sense? Yeah, so let's get back. Let me look at the time. I'm gonna finish up. I'll mention one more thing here and then we'll be done with this for today. So we had the synapse here. Let me erase a bunch of the stuff that we don't need anymore. Okay. All right, so we have the synaptic transmission. Action potential, neurotransmitter bind the receptor, message continues over here. Similar to what we saw in second messenger signaling with PDE. We have the same problem is we had the action potential saying, release neurotransmitter. Neurotransmitter is in the cleft, but now this signal is done. Once the signal is done, we want this neurotransmitter to be done. As long as the neurotransmitter is there in the cleft, it still binds to the receptor and creates some effect on the postsynaptic neuron. So what we need to do is have some way to clear the synapse of the neurotransmitter so it goes away, so the signal doesn't just keep happening. So we need to clear the synapse. Right? Does that make sense? After this happens, we need to clear the synapse or else the postsynaptic membrane is just going to keep getting triggered. So we need to get that neurotransmitter out of there. What's the most fundamental thing that's going to get the neurotransmitter out of there? Just from the stuff you already know about just little molecules floating around.
Diffusion.
Diffusion, exactly. So one thing that's going to help here is just diffusion. Right, you've got a bunch of neurotransmitter here, not a lot out there. It's going to naturally want to diffuse away. So partly just due to diffusion, it's going to start leaving. Another thing that happens is sometimes there are enzymes that break it down. You can have enzymatic breakdown. For instance, in acetylcholine synapses, often there is an enzyme called acetylcholine esterase that is actually anchored in the synapse and breaks down the acetylcholine. So it makes sure it doesn't just keep lingering. Like that's another I can mention, so one of the drugs that they use to try to help people with Alzheimer's is an acetylcholine esterase inhibitor. So acetylcholine is used in like different circuits in your brain involved with attention and things like that. So by blocking the enzyme in the synapses that breaks down the acetylcholine, then you increase the amount of acetylcholine that sticks around, and that seems to help these people with their, kind of maintain attention better, right? So there are, drugs that are prescribed that actually work by increasing the amount of neurotransmitter by inhibiting the enzymes that break down the neurotransmitter. What's I'll remember I'm forgetting the name of the drug. I'll remember the name of it in a little bit. And then there's one other important way to to clear the synapse. And, both diffusion and breakdown is wasteful. You make the stuff and it's either gone or you broke it down. But if you want to like go green in the synapse, reduce, reuse, recycle, re-uptake. And so another important way you can do it, if I'll do it in green, re-uptake. You basically have little pumps that bring the neurotransmitter back in so you can reuse it. Right, that makes sense. You released it, it's done its job, but now you just kind of vacuum it back up and are ready to go again. This is another part of the synapse that's targeted with a lot of drugs. Like SSRIs, like Prozac and stuff like that, fluoxetine. What do they do? What is an SSRI?
Serotonin reuptake inhibitor.
Yeah, so it's this a selective serotonin reuptake inhibitor. Normally, you release the serotonin, and then there are little pumps that pull the serotonin back in after, so you can use it again. The SSRIs block that process, and then you build up more serotonin in the synapses. And then there's a lot of other stuff that's going to happen after that. The reason they make you less depressed is not totally clear. And it's definitely not just about having amounts of serotonin in the synapse, right? Because you can take an SSRI and it takes like 3 weeks before people start feeling less depressed. So it's obviously not some direct effect of blocking the reuptake that's making you feeling less depressed. There's like something over the next 3 weeks, there's things that change downstream in your brain that ultimately are, resulting in the, kind of benefits. But at the most basic level, what they're doing is they're blocking the reuptake of serotonin, increasing the amount of serotonin in the synapses, but then that's going to cause a variety of kind of downstream rewiring and effects. So, but yeah, so whenever you think of SSRI, it's just blocking the reuptake, which is one of the ways to clear the synapse. Right, so again, when you're thinking about, this is kind of messy, clearing the synapse here, 3 main things. It can just diffuse out of the synapse, which is important. Enzymatic breakdown, important. Reuptake, very important. So